So one aspect of this that I've been exposed to a lot lately is something known as metagenomics. And metagenomics is the sequencing of entire communities. So what does that mean? So initially when people sequencing was difficult, and they tended to try to sequence just individual genes. And in doing so, they got the sequence of genes, and they might have been able to compare those genes between different organisms, different variants of the same strain, for example. You might be able to find a mutation in a strain if you sequence the right gene that contains that mutation and compare that sequence of that gene with the sequence found from the wild-type parent of that gene, of that organism. So that's one way of going about doing this. Then as sequencing became much easier, especially when you're working with viruses or bacteria which have relatively small genomes compared to, for example, the size of our own genomes, it became possible to not just sequence genes or regions of chromosomes, but to sequence entire genomes. And so you could sequence a genome and compare the sequence of that genome of the mutant, for example, that we used in the first example, with the sequence of the wild type, and actually determine what gene is mutated that gives rise to some interesting new phenotype that you're looking at. It's also possible to study the sequences of populations. So if you've got a population of organisms, especially microorganisms, which are relatively small, and this would have to be an isolated population. This would be something where you have just that organism and no other type of organism present. And so this is something that generally would be done under artificial circumstances, such as in the laboratory. But nevertheless, you could generate a population, for example, evolve that population over time, and then ask yourself, well, what kind of variation has developed in that population? And that can involve simply sequencing the population. So the population, you isolate the DNA from that population, you clean it up, you take that DNA and you sequence it, and by and large, you're going to, at every location on the chromosome, you're going to have a nucleotide that's present, but occasionally you'll see a different nucleotide. It won't necessarily dominate the entire population, but you will be able to see it. And as a consequence of that, you can infer that, in fact, the variation occurred at that point. And because you have the sequence of the organism you're working with, and that sequence ideally has been annotated, so you know where the genes are, you can infer where the variation is developing within that population. So populations can be sequenced. But again, having an isolated population is not necessarily easily accomplished, because usually there are other species around. And so this is something that typically you'd only see in the laboratory. So there's not a huge amount of demand for sequencing populations, but it is possible. It is also possible to sequence communities. So a population consists of just a single species of organism. A community consists of multiple species. And so anytime you go into any environment, you're going to find multiple different species all interacting together. Now it is possible, as I discussed earlier this semester, to sequence all of the DNA in an environment. And when you do that, and it isn't all the DNA, in order to sequence all of the DNA, what's known as the depth of sequencing would have to be too great. There's a lot of DNA out there. Instead, what you're doing with metagenomics is you're sequencing some representative sample of the DNA. That sample will tend to be biased towards those types of DNAs that are most prevalent with the environment. So those organisms that are most prevalent within the environment are going to be most represented in these metagenomes. Metagenomics allows you to go into some natural environment and ask yourself what kind of DNA is there, and from that you can infer what kind of organisms are there. And from that you can also infer, because you're sequencing the actual DNA, what kind of genes and therefore potentially what kind of functions are in that environment. Now, as an aside, in order to know what the function of a gene is, you have to actually do wet lab work. You have to actually do something that at least approximates biochemistry. And that is something that involves more than just DNA sequencing. So if we have a metagenome and we look into that metagenome, and we're able to say, aha, within that environment there is this function and that function and that function based on the genes that are there. Anytime a person makes an inference like that, whether it's a strong inference or a weak inference, that inference has at its basis somebody who previously did an analysis of an analysis of what that specific gene that you're talking about does. And that involves molecular microbiology, in the case of microorganisms, as done in a wet laboratory and very much has a biochemical basis. So we have metagenomes. So the idea of sequencing environments fairly rapidly turned into the idea that, in fact, bodies represent environments. Now, we've long known that there are flora, as we say, or microflora, that are associated with bodies. And in fact, there are normal microbiota that are associated with all environments. And those microbiota, the types of organisms that are present, are going to differ between different environments. And when you are creating a metagenome or doing metagenomics, all you're doing is you're sequencing the organisms that make up the microbiota that's found within wherever it is that you sampled that environment. And so it's no different to sample the environment that's associated with a human being, for example, and then sequence all of the DNA that is present, and from that infer what kinds of microorganisms are associated with that body that you took the sample from. Now, what organisms are going to be present are going to differ depending upon the location of the body that you sampled. So the skin, for example, versus the colon, for example, or the urogenital tract, for example. I mean, all these things are going to have different organisms present. But they're all going to have a relatively complex combination of organisms present. And we can infer what organisms are there by doing these metagenomes, by doing metagenomics, by sequencing all the DNA that's present. And to a degree, we can infer the functioning of the organisms that are present based upon what is known as sequence homology between the genes that we find in our metagenomes and genes that previously have been described, characterized using biochemistry in wet labs.